Today's webinar is designed to provide you with relevant insights into drug discovery and early research. Eugenio Fava will share his knowledge and experience on this topic, and I hope you will find the session informative, stimulating, and inspiring. We encourage you to actively participate in the webinar by asking questions and sharing thoughts, experiences, and checkbooks. We will do our best to answer your questions and make the webinar interactive and engaging experience. Again, we thank you for your participation and hope you find this webinar insightful and useful. Let's get started. The interview will begin shortly.
It is my great pleasure to welcome Eugenio Fava, who joins me today in the CELLIMA Talks series to talk about drug discovery. Hello, Eugenio. How are you today?
I'm fine. Thank you, Stefan, for the invitation here.
Thank you again for agreeing to this interview. I'm very happy that today's date worked out, and I appreciate that you took the time out of your busy schedule today. Eugenio, you are an enthusiastic and experienced scientist with a deep knowledge of neuroscience and drug discovery. Your research is specialized in neuroscience, and you have a strong interest in integrating high-content data with computational and research technologies to develop methods for application in cell biology and also in early drug discovery. Can you tell us a little bit about how you became interested in these topics and about your scientific background?
Yes, I'm a biologist in education with a PhD in molecular toxicology. And how to become interested in this, it was starting with microscopy and the digitalization of microscopy. So when biology started to become quantitative and not only qualitative, I understood that this was in the 90s, late 90s, that this was revolutionized the way that we look to cells in the microscope. And then on top of this one, when I was then later in my career, I understood also the development of laboratory automation that gave us the possibility not only to have quantitative biology, but to have scale up the experimental layout. And then I ended up at the early phase of the high content discovery at that stage. I was still in the UK when I was using one of the first cellomic machine that was sold at the time. And I moved then more and more in this high content and phenotypic approach, cell-based approach, and apply this more and more, not only in understanding disease mechanism, but also really for drug development and drug discovery. And so far we have now here in the ZTNI with a full platform for drug discovery that is fully automated with advanced image and data analysis. And we try to make use this one to tackle neurodegenerative diseases that are largely still not therapeutic or treatable in a sort of way. So we try to see what we can do there.
This is an exciting story. Thank you for sharing it with us. Brings me to my first question.
Mm-hmm.
What is the definition of drug discovery? What are the steps that are involved in drug discovery? And what are the advantages and disadvantages?
Drug discovery has many aspects. So drug discovery, in my opinion, it starts with understanding the disease and understanding the mechanisms that are key to the development, to the onset and development of the disease, and then identify targets that are critical and druggable, and then develop more insights for the future of the drug development afterwards. But drug discovery means also new chemistry, new technology as well. So how innovating technology can help drug discovery, and then thinking about all the different application from artificial intelligence now that we might touch later. But there is a constant development and also technological development from instrumentation to analysis of data. So those aspects are all included in drug discovery, in my opinion. So that's a really, but the key element is really understanding the disease and the mechanism of the disease. And especially, for example, in our field, neurodegenerative diseases are still challenging understanding the exact mechanism of the disease is not yet clear, especially, for example, take Alzheimer's disease. We studied the disease for more than 100 years, and still we have not really a complete vision of what is going on in this development of the disease.
What is the difference between drug discovery and drug development? Can you explain a bit about that?
Yeah, my opinion is that why drug discovery, as I told you, it's really understanding the biology behind, understanding the human biology behind, understanding all the different aspects, but also have a technology and innovation. Drug development, it's much more focused in bringing the drug to clinical trials. So that means for me, drug development is start after that you find your lead candidates and that you start to verify which lead candidates can become a lead candidate. And that means all the ADMET study, all the efficacy data and potency, and all the analysis. That for me is what is the classical drug development that take a long time, but is much more, if you want, technical and aim to really to isolate one chemical compound that can have a success in clinical phase. It's very critical as well because all the study that you do at this level, also for the drug development, will influence the output in the clinical trial. And we know that clinical trial phase 2 normally is the most critical, but definitely there are two different phases. The drug discovery, it is earlier, understanding all the disease, while the drug development it come at a later time point with all this ADMET study, toxicology, and everything else that is formulation, everything that is beyond the real drug discovery part.
Eugenio, what are the tools for meaningful drug discovery, and how can we efficiently find a new chemical structure that will successfully cure a disease, given the enormous number of compounds that need to be tested?
Yeah, I think that in the past there was this brute force approach where there were libraries of 3-4 million compounds that have been screened. I think this is largely now overcome by other techniques, of course. There are different aspects to have. There is one side chemistry. In my opinion, chemistry is somehow limited because the synthesis capacity of complex molecules is not definitely easy. Even if there are interesting development, for example, use of artificial intelligence and automated synthesis that now start to generate really interesting compounds that normally would not have been designed by normal chemistry, and this can overcome. I am a strong believer. I like very much to work with natural compounds because I think nature is power in chemistry that we don't have it, but definitely, of course, then some of the structure are very complex to replicate in the synthesis laboratory. But I like, for example, to work what we call semi-synthetic compounds, so backbone coming from natural compounds that can be synthesized and then used for screening. But the chemistry has a large component in the discovery because at the moment it is the key element to get the good candidate afterwards, and this chemical space increase is definitely a key. Other key elements are how relevant are the system or the assay that you use to your human pathology. More and more we drifted to use of human primary cells or stem cells derived cells. For example, we like to work a lot with the phenotypic assay because we think that even if it's difficult on the target, because it's a target agnostic approach, but it still resembles a system level of complexity in a cells. And I think that's important. A famous German scientist, Virchow in Berlin, some last 2 centuries ago was telling that every pathology is a cell pathology, and I do believe that it's still valid. At the end of the day, we have cell pathology that it just then go to the human pathology. So having the cell as a system, it's much more indicative. The challenge in this case then is to make the convolution for the target and the mechanism of action. But I think with the actual techniques, it's possible to do this kind of approaches, especially with the artificial intelligence and the data that are available now is really getting more and more feasible to have this approach. All this together might help to increase the output of the drug discovery, and not only the output, but also the translation to the clinical phases that is getting easier, hopefully. So you can discover side effect or efficacy level at very early stage, rather than getting at the clinical stage and then discover that your molecule is not efficient or is not does a side effect. So there are many things that can help in this bottleneck, twist the bottleneck there.
Do you think that drug repurposing can be used to shorten the drug discovery process?
Yes and no. So of course it is. So it is. It is being tempted by many of the company to repurpose drug, and I think that there is a value in doing this one, of course. But there are also obstacles. I mean, many companies do not have interest in doing this one either for IP issues. But definitely there is a value to understand the application of some of the drugs that are already in commerce to other pathology. Rather than repurposing, what I like, it is the new trend that also the authorities like the Food and Drug Administration are sponsoring more and more this real-world evidence in clinical trials, especially. So where you can derive information from different digitalization processes, from health records to clinical trials, and then get really input from the real world. So from drugs that are in use, or even non-drug, but just the behavior or other medication that patients can get in a different way, and then extract this information and try to understand if a given drug can be repurposed in a way that it's more significant. And I think some of the results are coming, and see the trends increasing. I see that this real world evidence is definitely getting more and more present in the drug discovery. And I expect it in the next year to become an essential part, at least at the clinical level, to understanding more what the drugs are coming and where they're going.
Eugenio, you're working in the German Center of Neurodegenerative Diseases, which is an academic institution. What is the role of academic institutions in drug discovery, and how does this role differ from the role of pharmaceutical companies?
I think, and I always believe, I mean, I will always working in academia. So my career has been always in different institutes, but in academia. But I collaborate with several pharmaceutical companies during and I like to have this part. I think one aspect it is academia should and must be focused more in what is drug discovery component. So and to innovate in there, and in the academia can innovate in different way, starting from the understanding of the disease. That's what we try to do, for example, for neurodegenerative disease here in Bonn, but also to innovate technology, to innovate at each level, from microscopy to artificial intelligence to develop, because that's the role that academia can do and must do, and the industry does, but it does in a different way. So in a sort of way, industry has more focus on the mission that the industry specifically have for the branch where they are integrating and interested in there. But academia can be done more freely and can also interact with more to disciplinary level. But a key role is really understanding the biology of the disease. Many of the diseases are still not completely clear. And that's our role. Or identify new target, for example. We have a shortcut in kind of targets. If you look at the Food and Drug approved drugs, I think that there is maximum 1,000 targets that are at the moment known. Despite a proteome that is probably hundreds of thousands of proteins, so there is still a lot of potential in there, and that's also another role that academia can have in identifying new targets. We started recently an initiative to increase, especially in CNS, the discovery of new targets, and innovating in all aspects of the drug discovery part. Of course, we play also a role in drug development, so we have all trained for doing that. But in this case, I see that academia can interact with the pharmaceutical world, because the pharmaceutical world is the professional work that knows all the regulatory aspects and has all the chemistry and everything. And I see the benefits to have more this translation of the discovery part to the translational part to industry. And that's also a role that the academia can play, and I think that is not yet fully utilized to have a more strategic interaction with pharma company.
Mm-hmm. When you're looking at the entire drug discovery process, what are the biggest challenges that the researchers are facing?
The different one is I mentioned already is the lack of targets, of course. So we are targeting always the usual suspects. So we need to understand more. But this goes hand in hand with what I say already, understanding of the disease. I mean, if you take cancer, oncology, it's very well developed. But if you take many other diseases, we still have really large issues in understanding all the molecular mechanism of action, the biology behind, and to understand this one. Chemistry is another aspect that I think you need to be improving to enlarge the chemical space in a more intelligent way that can be helping, and having models and assays that are representing the human biology. There are also other specific aspects. For example, in CNS, especially in neurodegenerative in particular, there is a lack of biomarkers largely. So in this case, there is the need to integrate more clinical science also at the early stage of drug discovery to understand the biomarker, to evaluate if the therapeutic intervention that you want to do is effective or not at the end of the day. Those are, I think, that the major area where there is at the moment issues that need to be tackled, and there is room to improve for sure.
Eugenio, you already mentioned a number of indications. Which indications benefit most from the different approaches to drug discovery?
Well, working in CNS, I think that there is a large margin to improve all the neurodegenerative treatments, from Parkinson to Alzheimer to Huntington. There is practically almost nothing. For multiple sclerosis, other treatment. I mean, multiple sclerosis, there is some things. In Parkinson, there is some things. Alzheimer, recently we saw some development, but still there is a large room to improve in the CNS area. And I think also the metabolic diseases is large improvement there possible, but also inflammatory component. I mean, inflammation is emerging more and more as a key central role on many diseases. This can go from NASH, liver disease, or to obesity and other things. And I think also in inflammation there might be a lot of room to improve. Oncology, I think, is pretty much developed. It's probably the most developed, but still also in there is immune oncology. There is a lot of space also there are many diseases that can benefit from increasing the drug discovery and drug development pipeline for sure.
Mm-hmm. Yeah. What is the most promising area of drug discovery?
I think CNS is still, in my opinion, a frontier, but also the one that offers more opportunity. That's, I do believe. And I think a CNS disease, not only on neurodegenerative disease, but many other aspects like chronic pain or depression or other disease, there is still room there to make really large benefit for the patients.
Mm-hmm. Mm-hmm. Coming from a scientific point of view, going to more technology perspectives now in our interview, brings me to the next question. What novel or emerging technologies are currently being used in drug discovery?
I think that at the moment, in my opinion, the one where there is largest innovation and largest opportunity is artificial intelligence and the application of artificial intelligence to data obtained for drug development and drug discovery. We have seen emerging in the last 10 years a lot of technology, and we will see more. and that's really start to be a very interesting area of development. Is already concretely there. There are some of the companies that that are already established that develop. I mean, I think about Recursion, Pharmaceutical, or Exsentia, like the two leading company, in my opinion, in this sector. But there is going to be more and more coming up in this field.
Though this is for you the technology which will have the most significant impact to improve the drug discovery workflow and the outcome?
It is one that is going to revolutionize the way we think about drug there for sure, because that will help us to understand what I said before, the disease, because we can have a look to integrating more information from clinical to experimental information. There are possibility to really think differently in this way. But there are also other technology. I mean, I think about the RNA. I mean, with the COVID, we have seen that the RNA therapeutic intervention is now a reality. So I think that there's been massive now use of mRNA vaccination. This is going to be also some things that revolutionize the way that we think about drugs. So we will not think only small molecules, but I'm absolutely sure that in a few years RNA will be applied for many applications, starting with oncology, but also other applications will emerge. And it's a very exciting time because there are different things happening in the drug discovery panel that will change the paradigm of how we think about drug discovery and drug development.
Which of the other technologies out there that are commonly used or novel technologies out there that are commonly used in the labs, in the drug discovery labs, are more nice to have, in your opinion?
Well, everything is nice to have. It depends on the resources that you have to use. But I mean, for example, the 3D cell models is now, I don't consider this one as a novel technology. It's already established, especially in oncology, but it's start to be also interesting in different aspects like neurobiology itself. But this can be and are already a reality. And it's always nice if it's possible to flank a model, a 3D model to a normal cell model. Even if the complexity increase and there are some caveats, still it is a nice tools in the toolbox, definitely. I think on the hardware and in general for microscopy, we have seen quantum leap jump in the last 20 years. Probably we are reaching plateau there, and there are a lot of technologies that are established. Single cell analysis is for sure nice to have. If you have the possibility to understand single cells level information is always very, very nice to have. There are many technologies that are available for really getting content inside your analysis that practically is unlimited at the moment. It's mass spec is getting more and more integrated also, transcriptomic itself. So there are many other tools that is nice to have. The core is, in my opinion, is always the cell models and the cell assay that you develop. Around this one, there are many docking technology that you can use, and it's nice to have around.
Eugenio, you already talked about artificial intelligence. What is the role of artificial intelligence in drug discovery? And what is the difference between machine learning and deep learning?
Yeah, that's artificial intelligence. It's very, I mean, artificial intelligence is a big container in containing machine learning, deep learning, and other statistical techniques. All of these, when I'm talking about artificial intelligence, I talk of everything from image analysis to data analysis and data integration. I mean, the first approach, for example, in eye content screening, if you have an eye content, it is the image analysis. So the deep learning, for example, has revolutionized the way that we do image analysis. Now, very complex image setup that a few years ago could not be analyzed, now can be analyzed, including also histopathological information, of course, that are very, very interesting information. But then it goes deeper in the analysis of the data itself obtained by primary experimental data, where you can get a deeper insight, association of different matrices is possible. But what I think artificial intelligence in general is really, really important. It's the integration of different information, ranging from, for example, overlapping the chemical space to the biological space. It's very demanding for human being, but it's very, it is possible nowadays to do with artificial intelligence, but also adding additional information like clinical information or more protein level information, and merging all this information together will show a different light on the complexity of the biology, and will also help in understanding how to deconvolve this complexity, to understand what it is the difference between a pathological status and a physiological status, and try to use this information to reverse the pathology towards physiology. That's what I think the artificial intelligence in general will do at the end, and is already there.
And this is also the way how machine learning and deep learning is using to improve drug discovery, in your opinion?
For sure, it is already happening, as I told you. So we see, I mean, for example, target identification will be one of the things that definitely will benefit from this kind of application. And then having more targeting means having more possibility to cure disease. That's in my opinion. It's happening already now, and we will see in the next 5 to 10 years an explosion on this direction, in my opinion.
Do you also think that machine learning and deep learning can be used to predict drug response?
To some extent, it's been already proven, yeah. Even if there are still caveats, but definitely there is a level of predictivity, and the more the machine learning and artificial intelligence method we develop, the better will be the prediction. What I think that is more necessary, especially from academia level, is to get a more common effort to tackle this problem. At the moment, there is a bit of disconnection, and there are interdisciplinary difficulties. I mean, you have to work from the biology, but also from the computer science, data science, mathematicians. There is the need to develop, for example, special curriculum in the university to have really this information merged together in a team or in a single person education. But it's coming, so for sure it will come, and yes, it will help to speed up the drug discovery. Absolutely, in my opinion.
And how can these applications and neural networks coming from machine learning and deep learning be used to study disease progression?
And this is the problem with the clinical information. So technically, it's possible to do it right now. The problem is all the regulatory and data privacy aspects from the clinical information at the moment are an obstacles if you want to integrate it. I mean, how to make a multi-site clinical trial and share information across different scientists to understand, for example, the clinical trajectory of Alzheimer's disease, it is a challenge at the regulation level. So I think that in this case, there is the need, and there is also the need to go and talk to regulatory affairs or to the authorities and try to find a common solution to, at the end of this, make benefit for the patients, because that's something that at the moment is an obstacle. So the regulatory, I understand that data privacy is important, but there should be a way to solve these issues so that more science can be done at this level. Because if we want to understand the disease, we have to work with patients. And actually, it's in the interest also of the patients to get a better treatment, and we should make a win-win situation out of this one. But this we need some discussion and some time to change regulation. But definitely, AI, artificial intelligence, is now making this possible. So there should be a discussion going on this for sure.
And I guess this is also true if you want to use this machine learning and deep learning neural networks to be used to improve drug safety and efficacy.
Absolutely, the same things is applying. Even if for drug safety, there are many databases already available. For example, there is drug adversary drug reaction database from the Food and Drug Administration that is available. But of course, it would be nice to have more information on the patients itself. And at the moment, it is limited. So we need to understand which kind of input we want to put in the artificial intelligence. So an artificial intelligence method is working always with the same principle. It is as good as the data that you feed to the system. The more data and the better the data, the better are the outcome. Huh? So I think everybody at the moment talk about GPT. This system has been trained with an enormous amount of data to generate still inexact, not completely exact output. And the same is valid also if you want to apply this method to drug discovery. The better the data, the more curated the data, the better will be the results of the system.
And what data is needed to feed machine learning and deep learning networks for an efficient drug discovery process?
I think that there are three different main areas of data, in my opinion. One is definitely the experimental data from the biology side. One aspect is the chemistry, and more chemistry data, quantitative data from chemistry structure to overlap the biology and the chemistry and the clinical data. So clinical relevant data from patients, from clinical study, but also from real world evidence coming more and more together. So that's the three areas where I see the most interesting overlap.
Eugenio, you already talked about the advantages of three-dimensional cell models. And you also mentioned that this is a well-established technology, but somehow these cell models have become a prominent focus over the last few months. How can the 3D models be used in drug discovery? And what are the challenges when using them, especially when you have to deal with miniaturized and fully automated applications?
Yeah, it is established technology, in my opinion, especially in oncology, has been extensively used. I mean, of course, there is an advantage is to have a 3D model that will resemble more physiologically a tumor, for example, rather than having a 2D layers on a plastic that is not exactly the most physiological model that you can have. However, in my opinion, any model has limitation and any model has advantages. 3D give this complexity of the cell-cell interaction in a more structured way, but it brings also complication for analysis, for example, very complex data sets to analyze with microscopy, for example. It brings also relatively complex physiological limitation. For example, there is no vascularization normally in a 3D model, so that means you get a very hypoxic core of the organoids, and this complicated the life. But still, what is good in a 3D model or a 2D plus model, what I say is that you can do co-culture. You can put different systems together. And this, of course, is benefit because then you start to get really close to the physiology, to what cells interact. I mean, things about the brain, if you use only neurons, is not a brain. A brain is much more complex. There is astrocytes, microglia, there are oligodendrocytes. So rebuilding a 3D structure is, in this case, an advantage. We have, for example, developed recently a blood-brain barrier model based on human-derived cells from stem cells that is resembling very much the physiological condition of the human brain. And that's also a model that is very important. I mean, we see now, for example, that there is the treatment for Alzheimer's disease with antibody that is becoming a reality. But we do know also that this treatment are caveats. For example, 1/3 of the patients develop edema. That means the blood-brain barrier has a play in this one. And now we can predict now, for example, can we use this model to predict which antibodies in which patients treat edema and what we can do to avoid that? Because, I mean, of course, if you want to treat a patient and you get the brain edema, is not exactly what you want. There are the advantages to using 3D models, so that's for sure. They get closer to the physiology. That's one of the advantages, in my opinion.
Do you see the use of these 3D models mainly in the early drug discovery process or better in later stages of the drug development processes?
My opinion is more in the late stage, yes, where you can really model more complex system to predict eventually adverse effects that are never good when you go to clinical trial, and also to predict efficacy. So that's I think where the 3D model, in my opinion, was promising. There has been recently, for example, a publication where 3D model organoids reimplanted in a rat brain have been shown to reintegrate within the brain, so to innervate and vascularize and get integrated in there. So 3D models of organoids can eventually become interesting also as a real medical application, so for regenerative medicine. And that's still at the beginning, but it will develop more and more, in my opinion, as well.
When we talk about three-dimensional cell models, I think everybody in the audience will have a different picture in her or his mind. And what do you think, Eugenio? Which cell models are available and which can be used very efficiently for drug screening?
Well, there are available many cell models. So that's, I think that every model has limitation and advantages. My personal opinion is that getting closer to human is important. We use mainly only primary cells, either from the immune system or stem cells derived cells from human material. And getting close also to the clinical, we get also material from patients, so either mutation or other things. And I think this is the advantage. So if I will be a young scientist today starting to make a career in drug discovery, I will definitely focus to human material and get close as much as to the clinic is possible, so to the pathology. That's my advice, definitely.
Yeah. Is this also the main difference between three-dimensional cell models and conventional two-dimensional cell models that are used in drug discovery?
I mean, yes, it is. Two-dimensional models are fast. That's the advantages. So you can, for example, use this one for identify hit candidates in a very fast way. My approach is always start as simple as it possible and then get a complication through the way. So our philosophy, my philosophy is always start with the easiest assay that it's normally a 2D assay that is stable and it's routinely used, and then get as soon as you start to discover more and reduce your hit candidates to an arrow, then start the complexity in the biology and getting more 3D models and other models that are coming later. Also, tissue-like models can be interesting, but complication of this complex system is not suitable for me for screening, for example, in my opinion. Even if in oncology has been shown that it's possible, but it's very demanding still. Is technically and is an effort to do screening in 3D models, definitely.
But if one decides to use 3D models in drug discovery, especially for screening, what are the factors that should be considered when developing three-dimensional cell models for this purpose?
Reproducibility. So it's the variation of this kind of assay, in my opinion, is still very high, and it is a very dangerous field. You have to be sure that your assay is fully validated to the extent that you can trust that you can repeat the same results across long spanning time.
Eugenio, you already mentioned patient-derived cells. What is the role of stem cells and patient-derived cells in three-dimensional cell models for drug discovery, especially when you compare them to traditionally used attenuated two-dimensional cell models?
I mean, that's getting closer to the pathology. So especially if you have a genetic disease, that's very, very interesting to see getting closer there. It gives you the next hook for the clinical phase in a sort of way. So you can really, but again, this goes together also with other tools like biomarkers or other things that you can develop. At the end of the day, it is the drug development in this case. It needs to take in consideration other aspects. So can you use, for example, the stem cells derived from patients to develop a biomarker that then reliably will give you the output of your clinical trials? And can you start to understand how you modify these biomarkers within a stem cells derived environment? Those are all questions that are in the drug development very important. And then can reverberate again in drug discovery as well. No? So that's which are the target that you want to eat? Are you eating the right target? And is these cells derived by the patients helping you in understanding the molecular pathway that you want to target? there are different aspects at a different level. But at the moment, I think it's much more in drug development and the use of the stem cells. It will come also back to drug discovery, of course.
Actually, we are at the end of our interview, as far as I can tell. I haven't taken any more questions on my sheet, Eugenio. Thank you so much for your time. I really enjoyed our conversation. I was fascinated by your exciting and engaging explanations about the drug discovery process, 3D cell models, and the use of artificial intelligence in pharmaceutical research. I wish you a good day and a successful week, Eugenio.
You're welcome.
I also wish our listeners and viewers a good day. Thank you for listening. Hope you enjoyed our conversation. Goodbye to everybody.
Then have a good day. Bye.
Thank you for joining us for today's online webinar. It was great to have you all here, and I hope you found it informative and helpful. Eugenio did a fantastic job of sharing his knowledge and expertise on drug discovery in early research, and I'm sure you took away some valuable insights from his statements. I really appreciate your active participation in the chat box and the great questions you asked. It made the webinar more engaging and interactive for everyone. Thank you again for your time and attention today, and I look forward to seeing you all again soon. Take care and have a great evening. Bye bye.